첫 번째 꼴찌야. 안 불러주니까. 문제를 무슨 거야? 근데 컵을 저었다 갖다 놓고
So as of now, we are facing a lot of biodiversity threats, and along with it is the increasing number of threatened species around the world. However, there is a very little knowledge on how we could possibly protect and conserve these species. And hidden within the forest canopies of the Philippines are the endangered Leccanopteris species whose survival depends on the delicate balance of habitat and biology. So, Leccanopteris species, also known as ant ferns, are actually epiphytic fern species that are distributed across Southeast Asian and Malaysian regions. So why do we call them ant ferns? So as you can see in the picture, they have this modified stem, also known as the rhizome. So it serves as domatia or home for ant species, specifically the stingless ants from the genus Erythromyrmex and Primatocluster. So there are actually 13 species in the world, and its center of diversity is actually in Sulawesi, of which out of the 13 species, they have 8 species, and of the 8 species, 6 of these are endemics. And in the Philippines, we have 4 known species of Leccanopteris, but now we will be focusing on only 2 species, Leccanopteris diparioides and Leccanopteris cyanoosa. So, for us, saving a species begins with answering 2 fundamental questions. First, where can it survive? And how can we help it thrive? So our research brings these questions together by integrating ecological assessment with in situ propagation to develop an ecosystem-based conservation framework for 2 endangered Leccanopteris species. So, although Leccanopteris cyanoosa and Leccanopteris diparioides are recognized as endangered species, we still know surprisingly very little about where they thrive or how we can effectively conserve them. So their survival depends on the specialized host tree or the phorophytes and the unique microhabitats and intricate ecological interactions. Yet both ecological knowledge and the practical propagation methods remain limited. So bridging these 2 gaps is essential for developing effective conservation strategies that support not only the protection of their natural habitats but also the restoration for long-term recovery. So to address these conservation gaps, our study pursued 4 objectives. So first, we examined the ecological requirements for Leccanopteris species by identifying its preferred host tree and the habitat characteristics. Second, we evaluated the host tree suitability and vertical distribution of this species. Third, we developed an in situ propagation protocol using indigenous culture media for both endangered species, and finally, we identified the media that best supported the gametophyte growth and successful sporophyte development. Here's a stepwise research approach to develop effective conservation strategies. So to answer the first question, where these ferns survive, we conducted an ecological surveys in specifically in Mountain Po Natural Monument located at the Camigan Island in the Philippines, which is biologically significant area recognized for its rich biodiversity and ecotourism potential. So it was declared as a protected area in 2004 and ASEAN Heritage Park in 2015. So with the survey, we have established 2 contrasting sites. We have first the open canopy site and we have the closed canopy forest. So for every individual encountered, we recorded its host tree, microhabitat characteristic, the canopy position, and the habitat suitability. And for in situ conservation, first we have collected fresh spores from different mountains in Mindanao, but before doing so, we have gathered all the necessary documents needed. Most importantly, the permits, the gratuitous permit from the DNR, and then the remaining spores we have stored in the oldest spore bank in the Philippines, located at CMU, Central Mindanao University, at 4 to 8 degrees Celsius. Next, we prepared all the primary media that we have used. So we used the grounded clay pots. So we also used anunutong, or the adventitious roots of tree ferns. Then we also used the garden soil. So after preparing all the media needed, we proceeded with the in vitro spore culture technique. So we followed the protocol by Amoroso et al. We modified it. Protocol by Amoroso et al. 2017. And all the cultures were maintained at temperature 20 to 21 with a relative humidity of 40 to 42% and LED light of 40 watts. So, although we recorded 15 host tree species, the population was concentrated on only 2 species. We have the highest habitat suitability index of 1. We have Hibiscus tiliaceus, followed by Pterospermum diversifolium of 0.52. So what makes a good host? So we have observed that a good host should be a mature tree, an open canopy, with rough bark, and a moderate branching. So host selection was strongly influenced by the tree architecture rather than the tree identity alone. So the rough bark traps the spores, retains moisture, and accumulates organic matter, while moderate canopy openness provides enough light without excessive drying. So in other words, the fern selects the habitat before it selects the tree. So where exactly does it grow? So regardless of whether the forest was open or closed, individuals consistently occupy the basal and the inner canopy. So these positions provide a balance between light availability and moisture while accumulating organic debris essential for epiphytic growth. So the tree height itself was less important than the microhabitat created within the canopy. So how is the population distributed? So we did compute the variance mean ratio. So the population exhibited a strong aggregated distribution. So instead of spreading evenly throughout the forest, individuals clustered on only a few suitable trees. So that's the Hibiscus tiliaceus. So this means that using only a handful of E4 could disproportionately reduce the entire population. So for the second question, whether we can actually conserve it or grow them outside their natural habitat, the answer is yes. So here we have the first documentation of the development for this specific species of ant ferns. For Lichanopteris diparioides, we're able to germinate them, and we have reached 100% of germination all throughout the treatments. And the spores germinated within 3 to 7 days, depending on the treatment. And we have observed the glycinia type of germination and the Granaria type gametophyte development. So these species have chordate phalloid gametophyte, and it has a funnel-shaped antheridia that appeared 24 to 39 days upon sowing. We also successfully documented the other species, the early development of Lichanopteris cynosa. So it grows 2 to 5 days after sowing with a 100% germination rate. It exhibits the Bitaro type of germination, and along with diparioides, it also shows the Granaria type of gametophyte development, and both species have chordate phalloid gametophyte. However, the heart formation or chordate formation of Lichanopteris cynosa is earlier compared to diparioides. And it has a barrel-shaped antheridia that appeared 34 to 44 days after sowing. So for the best culture media for the gametophyte survival, we found out that Lichanopteris diparioides preferred the treatment with mixture of anunutong, or the tree fern with grounded clay pads, and it reaches 95.36% of the metaphyte survival. So this mixture actually provides balanced moisture aeration and substrate stability and moderate nutrient availability. So the improved survival of the Canopteris diparioides in that makes aninotal and clay medium may reflect adaptation to the natural conditions encountered on host trees. So in the field, the spores develop on bark surfaces where decomposed organic matter, mineral particles, and humus accumulate within the bark features. So the mixed medium likely recreated these microhabitat conditions more effectively, thereby enhancing the long-term gametophyte survival. So for the other species, the Canopteris sinuosa, it best grow in garden soil, in mixture of garden soil and grounded clay pads. So that mixture provide stable moisture, and it improves rhizoid anchorage, and it releases gradual nutrient, favorable conditions for fertilization and embryo development. So these contrasting responses suggest that each species has distinct physiochemical requirements for successful gametophyte establishment. For the best culture media for sporophyte survival, both species develop well in both in a mixture of ground clay pads and garden soil. So we have created a systematic approach to conserve forest resources through propagation, production, and long-term sustainability. So research updates, as of now we have hundreds of cultured plantlets ready for acclimatization and transfer. So this is for the summary conclusion. So that would be all.